Okay, so this is the pre-lecture for occupancy modeling, and I'm going to talk a bit about species mark recapture studies and then also species distribution models as they relate a bit to occupancy. So starting with mark recapture studies, that the problem we're trying to resolve with mark recapture is might be how many of some species X are there, where do these individuals spend their time, what's the size of a home range, do males or females disperse, where do X migrate to and from, and so it's all the kinds of things where we have animals that move around and we want to know where they spend their time. And this is going to be developed further in habitat occupancy, but unlike plants, where mark recapture study is pretty easy, you go out and mark a plant, and then you come back and you see it exactly where it was last time, the next time you go out. Animals move about, and hence they're not likely to be exactly where we find them last time. Plus, animals hide, and hence, even if they are pretty close to where we found them last time, we might go out and then not see them. And so there's a detectability issue that Aviv will talk about tomorrow. But simple mark recapture studies, there's a Lincoln-Peterson estimator, very, very simple math. It just is this idea that if you went out and marked a fraction of individuals, and so, or marked some number of individuals and threw them back out into the population, that the fraction of those marked individuals over the total population would be the same as the fraction of some number of individuals you capture at a later time that are marked over the number that you capture, if these populations meet some assumptions. So put the fraction of individuals in a sample that are marked should be an estimator of the total number of marked over the whole population. And so if we mark 10 individuals and go back later and capture a sample of 20 individuals, and 2 of those 20 individuals, or 10%, are marked, then we presume that we have marked 10% of the original population, and that we could estimate the total population size of or the size of that population. Of course, this assumes quite a lot. It assumes that there's a closed population, no births, no deaths, immigration, emigration. No individuals lose their marks or gain a mark accidentally. Individuals are well mixed, and hence the capture of them is random. That means that individuals don't exhibit behaviors that change the likelihood of them being captured after they've been captured once. So individuals aren't trap happy or trap shy and things like this. Well, some of these assumptions are pretty steep, and going out, measuring, marking some individuals once and going back and capturing them again is not a very robust way of estimating them, and so there have been considerably more detailed and complicated methods that are basically working on the same fundamental principle, that the number of individuals out there marked is over ones that aren't marked is some estimate of the frequency of those individuals. And so there's Jolly-Siever models that then model an open population. There's the program mark, which is software to estimate, but can also estimate the number of individuals entering and exiting a population, possibly even bias and trapping, based on repeated capturing and capturing and capturing and capturing organisms. So home range mapping gets at some of the same issues when territoriality stomps on this well-mixed assumption that if you have animals that are defending territories, then they most certainly aren't well-mixed, and you have to go about doing these things in a different way. So it's just a few steps forward to putting radio collars on animals and then making home range estimates to find habitat quality and so that you can do things like protect habitats that capture the entirety of a life cycle. So, for example, tiger salamanders, we know we capture them when they're in vernal pools here, but we don't know that much about what they use and when they leave those ponds and hibernate for the summer. Well, it turns out that they might go quite a distance away in the grasslands and then go more or less dormant. Well, if you don't capture any of that grassland habitat around the vernal pools, you aren't likely to have a population that can persist through time. can also imagine then using these kinds of data for minimum viable population estimates based on saturating territories. How many individuals of how many grizzly bears could you have in Yellowstone based on the kind of the way that things move around and interact with one another. One other step up in technology and constraints on what organisms you can do this on are things like GPS tracking and things like finding migration pathways. And with GPS tracking, we've now know a whole lot more about some of these massively migrating species that move from the Arctic all the way down to the southern hemisphere and sometimes fly for 48 hours straight and lose half their body weight. And really interesting stuff based on GPS tracking. But we might also think about then getting movement data and connectivity and thinking about how patches are connected together and what this says about the potential for a population to persist. And we'll talk a bit more about connectivity later. But basically the idea here I'm trying to capture is that any kind of data that allows you to go out and mark and recapture studies has some of the same issues that we're going to talk about with occupancy in thinking about how detectable is a species in its environment. How likely are we to find it and be able to say something about that population then. And now I can switch over to species distribution models. Now species distribution models also share a similarity with habitat occupancy in that they're both occupancy models and distribution models are trying to make a geographic statement about what represents a good habitat for a species to persist in. And they're fundamentally at different scales and using different kinds of data. The distribution models are basically using museum data or collections data, places where we've seen things before, to make predictions about where things might otherwise be, or predict where they might be under changing environmental conditions. And as such, it's not trying to estimate the likelihood of finding something when you actually go out and look for something. Whereas occupancy models are generally done at a much finer scale, and they're looking at questions like, well, does this shrub density or does that shrub density or this other grass density, do they all constitute equal habitat quality for this bird species that might move across the habitat patches and use all of those things. And so you might get a better idea through habitat occupancy modeling what distribution of land cover types or land cover states would you want to protect. Whereas distribution modeling is much more about ranges. But then it goes back to Grinnell and the early 1900s where the definition of a niche, and there's environmental niche models, and right around 2000 you see this amazing flourishing of this literature with Antoine Gusan and Zimmerman in 2000 publishing this paper, Predictive Habitat Distribution Models in Ecology, and I'll show later the absolute explosion of the literature following this sort of work. In 2002 in Nature, this Raxworthy went out and published a paper using these methods and then finding a variety of new reptile species in Madagascar as a consequence of predicting where you might find them. And so this was a big feather in the cap of distribution modeling for its utility. And Town Peterson and Stockwell and others published a number of papers looking at new techniques that you can use to to model distributions of species. From 2002 to 2004, sorry, that's a little confusing on that slide, Jane Elith and Catherine Graham organized a working group that Gasson and Thuler and Moritz and Peterson and Simon Ferrier and others that are sort of notable in this area participated in, as well as our own Robert Hymans here. And they published now a very famous paper in 2006, Novel Methods in Species Distribution Modeling in Ecography, that Jane Elith was the lead author on. And about in the same time, 2006, this guy Phillips, who works at one of the research institutes from Bell Labs or something, published Maximum Entropy, a software package that will help people do distribution modeling on presence-only data. So there's been a number of advances right there in the middle of the 2000s. So what do people use distribution models for? New species discovery, to prioritize sites for surveys, prioritize sites for protection, And probably more commonly and more importantly now in the future is to investigate the consequences of changing environments like land use disturbance and climate change on the potential habitat or locations of species. So what does it do? It uses distribution occurrence data like this, hypothetically the stuff up in the upper right, along with a lot of information about the landscape, and to then use these landscape predictors. These might be climate or land use, or elevation, topography, anything that to predict where you find occurrences. And so this is a diagram from a annual review done by Jane Eliff that I'll post up on the site, where you have map species data and you have rainfall and roughness and southness and much of environmental attributes, and then you can put the landscape into some sort of multi-dimensional model. This is the gray points, and then ask where do those occurrences actually occur in there, and then map and then give everything in the landscape a score based on the probability of occurrence, and then remap that back onto the landscape to look at where you think things might be in the and for what reasons. And then you can also change those future landscapes and ask where the climatic parameters or that rainfall might be under a future climate model, and then which suggests where the species may pick up or lose habitat in the future. So naturally, there's lots of different methods to do this. There's maximum entropy, multivariate adaptive regression splines, regression trees, general linearized models, genetic algorithm, rule sets for prediction, GARP, GLIM, MARS, Maxent, and they give different answers if given the same data. And so one has to do this with some caution, because you can get different answers to the same questions using the same data, using different methods. But using climate change as an example, people are doing this in abundance now. The lower right here is the distribution or projected distribution of Joshua Tree, where green represent areas that might become future habitat, red are areas that are habitat now but are likely to be lost, and the orange and yellow, which you can't see, are those things that where the current distribution may overlap with the future distribution, and this has given rise to thinking that things like Joshua Tree are in deep trouble. But also using these models to try and predict diversity into the future, predicted future species richness, or the changes of ecosystems. You can stack those up and do all sorts of projection of future change. Kassan et al. In a working group in 2013 published a paper looking at this and how you use translators to bring things like species distribution models into the decision place, with thinking about developing distribution models and then using them to solve conservation problems. And they identified 4 different areas: biological invasions, identifying critical habitat, reserve selection, and identifying places for translocation as places where distribution models may help clarify a decision context. And they put this within a structured decision making sort of framework. So it's SDM, structured decision making, or SDM, species distribution modeling for SDM, structured decision making, as it were. And this is problem identification, defining objectives, possible actions, consequences, trade-offs, and decisions. So that's that PROACT acronym there over on the row headings. But people want very fine-scaled information, and here's a paper that we did. Oh, I'm at 14 minutes. I'm going to have to stop. I only have a few slides to go, but it's going to take me a couple extra minutes. So I'm going to end this one and start a new one.